I'm Anna Grenfell, the Community Facilitator Registered Nurse for the ME CFS Group, and it's great to see so many of you that I've spoken to before, either on the phone or by email, come to this event. So we're a non-profit organisation providing services to those with ME CFS in the wider Canterbury region, and I'd like to introduce Dr Eiren Sweetman of Otago University. Dr Sweetman has been researching the biology of ME CFS since 2011, and in May 2018 graduated with a PhD focusing on this topic. She's here today to share the results of her research as well as discuss key findings from international research. Our hope is that you will come away from this talk with a better understanding of ME CFS and apply what you have learnt to your clinical practice. Also,
thank you. Good evening, everyone, and thank you very much for inviting me here to speak to you all and share the research that we've been doing down at the University of Otago, and also some of the international research that relates to ours, and also that I personally find very exciting for our future growing understanding of ME CFS. So, yes, this works. First, a little bit of a background on ME CFS. I'm sure most of you are familiar with the illness. It's a severe debilitating fatigue illness. It lasts for over 6 months. It's a chronic illness, and often it's a lifelong illness. It often follows an acute viral infection as an initial trigger or a sort of extreme stress event. And flu-like symptoms are common, along with a range of, sort of whole body effects, cognitive effects, brain fog, muscle pain and joint pain, sort of orthostatic intolerance, and severe cases are bed-bound. The likelihood of a full recovery from ME CFS is poor, so 25% approximately will stay in this acute phase of the illness, the very severe symptoms, and only 6% of people fully recover from ME CFS, and even that is a little bit up in the air as to whether they really truly recover to their normal sort of functioning state. So it affects actually a very large proportion of people. It's estimated up to 2.6% of the population worldwide, and that's about 20 to 25,000 people in New Zealand suffering from ME CFS. So it's an illness that deserves our attention. One of the major issues with ME CFS is the question of how do we diagnose it? How do we accurately diagnose it? To date, there is no specific clinical test available for the illness. There's no diagnostic biomarkers. A single causative agent for ME CFS is currently unknown, and the disease pathophysiology is still relatively ill-defined. So our research down in Otago is aiming to tackle this issue of diagnosing ME CFS. The approach that we've taken is based on precision medicine, which is originally known as personalised medicine, and it was used in cancer treatment. So it was the idea of just looking at one individual, doing a lot of comprehensive molecular analyses on that one person to find the best possible treatment for them as an individual, rather than giving them a sort of what you would give to the general public. And so it's now expanded into diagnosis and prognosis of illnesses, particularly cancers, but also of rare illnesses. So we have approached ME CFS by gathering a group of 10 ME CFS participants initially. We've now recruited more into our study, but our initial analyses were all done on these 10 very well characterised patients. They were all diagnosed by one expert ME CFS GP, and she uses the 2003 Canadian consensus criteria, which is generally agreed upon by researchers internationally as the best diagnostic criteria to use for research purposes. We had a higher female to male ratio in our study group, which is seen in ME/CFS, and 8 of our 10 were said that they were in the acute stage of illness when we took initial blood samples for the study. So this is an example of a precision medicine approach to ME/CFS. We isolated plasma and peripheral blood mononuclear cells from our patient samples, and we analysed a range of different molecules from these two things. We looked at cytokines, microRNA, the transcriptome or the expressed RNA in the cells of these people, the proteome or the expressed proteins in peripheral blood mononuclear cells, mitochondria, which has very recently become an area of interest in ME/CFS, and the new Seahorse analyzer technology allows us to really delve into what is going on with mitochondria in the illness, and also epigenetics, which is the modification of your DNA influencing gene expression. So first I will talk to you a bit about our proteome results, which is probably our most recent and I think exciting results that we have from our study. To remind you all, I will be talking quite a bit about the mitochondria, the energy powerhouse of the cell. So this organelle in your cell provides all, or the majority of the energy your cell needs to function for you to live. And it's in this form of this ATP molecule. The mitochondria, not only the ATP produces the powerhouses of the cell, responsible for converting the air we breathe to energy. They also are involved in a number of other very important pathways. So in calcium regulation, inflammation, they are involved in inflammatory signaling, and they also are influenced by inflammation, senescence, cell death, and in producing reactive oxygen species, which are a kind of a double-edged knife in that they are a signaling molecule, but if levels of reactive oxygen species climb too high or remain too high, then they can cause damage to your cells and to your DNA. And again, this is a little bit of a busy slide, but it's talking about this ATP generation pathway. So again, just a reminder, because I'll be talking a bit about our results relating to this in the next slide. So your food is broken down into carbohydrates and fats to form this acetyl-CoA molecule, which enters your citric acid cycle and is broken down further into NADH and FADH2. Both of these feed into the electron transport chain. There's 4 or 5 complexes in the inner membrane of the mitochondria, and they pass electrons through all of the complexes via some sort of cofactors, CoQ10, cytochrome C, and in doing so they pump hydrogen protons across the membrane, and so there will be an increased concentration of protons on this side and a deficiency on this side, and as a hydrogen is pumped back by complex 5, the ATP synthase complex, an ATP molecule is generated. And oxygen is also consumed in this process, so it's a form of aerobic respiration. So these are our proteome results, and I'm not going to go through every individual protein that we found differentially expressed in the ME/CFS group, but I wanted to highlight these things. So we found a number of dysregulated and primarily increased protein expression in the citric acid cycle, in complex 1, in reactive oxygen species regulation, in the ATP synthase complex 5, and also in complex 3 a little bit. Another thing I didn't mention before, but complex 1 is the primary producer of reactive oxygen species in that electron transport chain. So it was interesting to see complex 1 and reactive oxygen species regulation coming up as affected pathways in ME/CFS. why this got me so excited initially after seeing this very clear sort of biological pathway in the proteome analysis, we also recently read Dr. Paul Fisher's paper. He's a researcher in Australia, and he's recently come into ME/CFS research. He published a paper just last month that whose results agree very highly with ours. So he took 51 ME/CFS patients diagnosed with the same diagnostic criteria as our study, and he found a deficiency in complex 5, that ATP synthase. It was worse at making ATP than healthy controls. And he also found that this deficiency was being compensated for by an upregulation of those other 4 complexes in the electron transport chain, in particular complex 1, which we also saw in our proteome study. And he saw an increase in membrane transporters and in enzymes involved in fatty acid beta oxidation and in the citric acid cycle. So he has formulated this hypothesis to explain ME/CFS, to explain the post-exertional fatigue, which is a key symptom of ME/CFS, that this deficiency in complex 5 is being compensated for by these other pathways earlier in the ATP synthesis pathway in resting cells, in sort of steady state cells, but this will leave them unable to respond to any additional stress, so exercise or emotional stress that is applied to the cell, because these compensatory mechanisms are already active and in place. So this is a little summary of what I've been trying to explain. So what do these 2 studies suggest? That mitochondria are dysfunctional in ME/CFS. The ATP synthase complex is deficient in producing ATP. This leads to a compensatory increase in complex 1 in particular, and the other electron transport chain complexes in citric acid cycle proteins. If complex 1 is increased and overactive, then more reactive oxygen species will be generated, and this will lead to oxidative stress in the cell. And that is again something that has come up in our studies and in other studies of ME/CFS. Complex 1 is also involved in the formation of the cristae of the inner membrane of the mitochondria, these little loops and folds like a brain. And if there's an imbalance in complex 1 expression and these other complexes, then cristae formation is affected, and cristae formation has been shown to, if it is affected by this dysregulation, to also impact on mitochondrial function and this ATP production. So it's a very clear pathway to explain this post-exertional fatigue that ME/CFS people experience. Moving on to the next study, obviously we're interested in mitochondria, and we now have the seahorse technology down at the university to analyse it further. We had 3 sort of main questions we wanted to ask when we were looking at mitochondria. We wanted to, first I'll talk about seahorse, then I'll talk about our questions. So the seahorse analyzer, it's a mitostress test we use. So it's a test on live cells, put them on a plate, put them into the machine, and then we can inject different reagents at different time points in these live cells that affect different parts of the electron transport chain and allow us to measure how much oxygen is being consumed to produce ATP, the maximal respiration capacity of the mitochondria, any non-mitochondrial respiration, any proton leak, and the spare respiratory capacity, which is this difference between ATP production and the maximal respiration in the cells, in our patients and our controls. So we wanted to generate a bioenergetic health index for our patients. And a bioenergetic health index is essentially a number, and it's a measure of healthy mitochondria with very good reserve capacity versus some sort of bioenergetic dysfunction, which is majorly reducing the ability of the mitochondria to produce energy, this reserve capacity. So it's a scale essentially. And we wanted to eventually, and we still do, want to give patients a bioenergetic health index number to take to their GPs, to take to their workplaces as proof of their deficiency in energy and metabolism. This is an example of a severely ill ME/CFS patient compared to 2 controls. So this is relating to this figure here, but at a real experiment. So these are our 2 controls, very similar in their mitochondrial function at every stage after injecting the different reagents. And our ME/CFS, severely ill ME/CFS person is significantly reduced in their mitochondrial function at every point. CoQ10 is a molecule I mentioned that's involved in the electron transport chain, and we were interested in whether there would be a difference in CoQ10 levels that could be an explanation for why mitochondrial function is dysfunctional in ME/CFS. We found that there was no difference. We looked at plasma samples and peripheral blood mononuclear cells, the levels of CoQ10 in all of our patients and controls, and we recruited a new cohort as well for this study. But this is kind of expected, and we saw this as well with our Proteome study, that there was no difference in CoQ10 level. And we've seen now with our Proteome study that the deficiencies can be explained by the increased expression and the deficiency of the ATP synthase. So this was done a little bit earlier to our Proteome study, but worth mentioning. This was initial bioenergetic health index numbers we generated from frozen peripheral blood mononuclear cells from our patients and controls. And the results do suggest a lower BHI number for our patients. The difference is not statistically significant, however, due to the smaller sample size. You generally will need to replicate this on a greater number of patients and controls to generate that significance or find if there is a real significant difference between the 2 groups. But it does look like, to me, that there is a lower BHI number. In relation to CoQ10, we were looking at MitoQ, which is a CoQ supplement. It's a dietary supplement, and it has a tag on it that targets it directly to the mitochondria. So we are doing these studies at the moment. This is just an initial result, and it's just looking to see whether MitoQ is effectively taken in by cells versus just CoQ10 without the tag. And this one line here is MitoQ at a 5 micromolar concentration incubating our cells with this supplement. So over this 4-hour period, it does look like it is being taken up by the cells. So we're now going ahead with studies with our patient and control cells to see whether it will ameliorate any of the dysfunction that we see in mitochondrial activity. So results to come. I will now talk about our Transcriptome study. Sorry, there's a lot of studies here to talk about, a lot to share. Our transcriptome analysis was published at the beginning of this year in an international journal, which was fantastic. We found that there were 27 gene transcripts increased and 6 decreased in our patient group compared to controls. And the 3 most highly differentially expressed genes were these 3, IL-8, NF-kBIA, and TNF-AIP3. Those are their full names there. They are all anti-inflammatory responders to TNF-induced NF-kappa B activation. So they're all trying to dampen down inflammation in the body, suggesting that there is inflammation to begin with. And this is some figures just illustrating the difference that we found. We then looked at the differentially expressed gene transcripts, and we looked to see whether there are any functional interactions between them, whether they were all involved in the same biological pathways that could explain the symptoms that we see in ME/CFS, and a lot of them were. So these lines here indicate functional interactions between the genes in the group that we analysed. So there were a lot of different interactions and a lot of different common biological pathways. We did a larger ingenuity pathway analysis, which looked at a wider group of differentially expressed genes. Initially, we just looked at the most significantly differentially expressed genes. This was with differentially expressed genes with a p-value of less than 0.05. So again, more pathways came up that were of interest. So the production of nitric oxide and reactive oxygen species. Oxidative stress again came up, which was very interesting. And in summary, we found these 3 genes, all related to inflammation, all increased in our patient group. And our pathway analyses detected effects on immune and inflammatory pathways in the cellular stress response and oxidative stress, in the circadian clock function, which makes sense as a lot of people with ME/CFS have very disturbed sleep patterns, and in metabolism and mitochondrial function. And as I said, these results were published in the International Journal of Immunopathology and Pharmacology in January of this year, which is, it's always great to get your research out there for people to read. Briefly, I will talk about our epigenetic results. This is, again, research that's being done this year, just being completed by our master's student in our lab. So we've just got some very initial results from her. This is a little explanation of epigenetics. We were looking at DNA methylation of DNA sequences that affects whether that gene will be expressed or not. An initial study our master's student did found that there was a difference in methylation patterns, particularly in the younger age groups of our ME/CFS patients. And she found a number of mitochondrial-related epigenetic changes. So these are just a few of the affected genes that she found that have got a lot of methylation groups attached and will have their expression affected. Now on to a slightly different topic, talking about the neuroinflammation symptoms seen in ME/CFS, because that is a whole other aspect of ME/CFS that we haven't covered so much with our metabolism results. At the end of 2018, my supervisor, Warren Tate, and another student of his, Angus, published a paper. It was a hypothesis paper on the stress centre, the hypothalamus, and it was theorising that this could explain a lot of the neuroinflammatory symptoms seen in ME/CFS. This is leading on to another little section that I'll be talking about relating to stress and the hypothalamic pituitary adrenal axis. So the HPA axis prepares the body to respond to stress. It is the stress response axis in the body. When the body is under stress, the HPA axis can suppress non-important it can suppress growth and metabolism. Cortisol is released, which primes the immune system for action. And under chronic stress, this high cortisol will actually drop to low cortisol, and then you get sensitivity to cortisol. So very small levels of cortisol can produce the same big effects that high levels of cortisol would. So ME/CFS shares some symptoms with this high stress, chronic stress that you see with cortisol. Cortisol can explain those aspects, but it cannot explain the neuroinflammation that you see in ME/CFS. The paraventricular nucleus is in the hypothalamus, and it is the stress centre of the hypothalamus. It contains these corticotropin releasing. It contains 2 receptors which both respond to corticotropin releasing hormone. There's 2 receptors in low stress states. Receptor 1, CHR1, leads to a decrease in serotonin. In high stress states, receptor 2 is expressed on the cell surfaces, and that results in a large increase in serotonin. And they act as an on-off switch, so only in high stress states will this receptor 2 be present, and normal low stress states receptor 1 will be present. So they are internalized in response to stress. If you are become stuck at this receptor 2 stage, then serotonin will be continually produced, and you'll get this increase in serotonin, this abnormal increase and consistent increase in serotonin. And this can actually lead to many of the symptoms that you see in ME/CFS. Increased pain, cognitive dysfunction, sensitivities to light and sound, sleep dysfunction, and by stimulating other neurotransmitters, it can also give you indirect consequences. It can affect your mood, your appetite, your gastrointestinal functioning, your heart rate. So a number of things which are related to ME/CFS. There is a small company called Cortene which has just started a clinical trial with a drug related to serotonin. So at the moment they are basing their trial on the hypothesis that they have come up with to explain ME/CFS. There's limited research showing that there's increased serotonin and cortisol in ME/CFS, but the hypothesis does explain a lot of the neuroinflammatory symptoms, so they're going ahead with this phase 1 and phase 2 trial at the moment, and we'll see where that leads. They hypothesize that this receptor 2 gets stuck, and you get this increase in serotonin, and this increased sensitivity to serotonin. So even with small amounts of stress, you get this big increase in serotonin, you get these effects that cause ME/CFS. And they have this drug which binds to the receptor 2 and overstimulates it, which will cause it to be internalized into the cell and switch off this increased serotonin pathway. So as I said, we'll see where this goes, but it's interesting that there's a lot of different research out there that's pointing to possible explanations for ME/CFS. Relating to serotonin, I'll talk to you now about some international research that's going on, and this is something that has come up in this year that is very interesting, I think, and related to what I've just been talking about. So Dr. Rob Phair, he works at Stanford University. He's proposed this metabolic trap theory to explain ME/CFS. So he was an engineer, now into ME/CFS research, but he took his engineering mind and has applied it to ME/CFS and come up with this biostability sort of explanation for the symptoms. So he surmised from the fact that there are frequent epidemics of ME/CFS. So after a triggering event, up to 25% of people will go on to develop ME/CFS. That there must be a predisposing mutation to cause this susceptibility to ME/CFS. And it must actually be quite a common mutation for so many people, the subgroup of people, to go on to develop the illness. So he started searching the genome sequence for mutations in genes that could produce this biostability effect. So biostability is essentially two possible states your body can be in, healthy or unhealthy, depending on the activity of this gene, this one gene, or the gene that, the pathway that gene is involved in. So if that gene is broken, then you will go from a healthy state to an unhealthy state, and it's like a on-off switch again, a switch possible between these two states. And he actually did find several common mutations in IDO2. To explain IDO2, IDO2 and another protein, IDO1, both catalyze the same step in the degradation route for tryptophan. But IDO1 is inhibited at high concentrations of tryptophan, whereas IDO2 will keep working. So if there's high concentrations of tryptophan, IDO1 will stop working in this degradation pathway, but IDO2 will continue to degrade tryptophan and stop the levels of tryptophan from rising too high. But if the IDO2 copies are broken in ME/CFS if they have these detrimental mutations that stop the protein from being expressed and stop it from doing this key first step in the breakdown of tryptophan, then your tryptophan levels will rise too high. And tryptophan, one of the other pathways that it can go down is actually, it will be made into serotonin. So again, you would get the same high levels of serotonin potentially happening and the effects, the symptoms relating to that. So that is the IDO metabolic trap. It's a very interesting theory to explain ME/CFS, and he's now sequencing as many patients as he can get his hands on to see whether they all share these mutations in the gene. Another very promising piece of international research is the Nanoneedle diagnostic test, which is being explored by Ron Davis at Stanford University and his team. So this was sort of coincidentally found. They were using the Nanoneedle technology to try and replicate the post-exertional malaise that ME/CFS people feel. So they had their cells on there, and they added a high salt stressor. So if you increase the osmotic stress on the cell will start tearing through its ATP reserves, and you will replicate the stress, this exercise stress on the cell. And he actually sort of found this electrical impedance resistance sort of accidentally as he was doing this study. So this is 20 ME/CFS cases and 20 healthy controls, and all of the patients have this sort of anomalous electrical impedance response measured by his nanoneedle. So he's again pursuing this very rapidly. His son actually has ME/CFS, so he's a very motivated researcher, and he has a very big team backing him at Stanford, so it's great. It's what we need for ME/CFS research. And he's already exploring 2 drugs which have been shown to reduce this response when they measure it with the nanoneedle biosensor. There's Copaxone, which is used in treating MS relapses, and a new unapproved drug, SS31, which actually targets the mitochondria, so mitochondria come back. At the moment, they don't have a clear explanation of what is causing this electrical impedance, but as they say, a diagnostic test doesn't always come with a clear explanation of why you're seeing the difference. You can still use it potentially to help diagnose ME/CFS. So that is a very exciting piece of research. The last thing I would like to talk about, in New Zealand, it's often quite difficult to successfully obtain funding for research, so we do have to look overseas for a lot of our collaborations and I guess hope in pushing forward ME/CFS research, although we do our best here. So there's excitingly a really growing collaborative research happening, particularly in the US, where researchers such as Ron Davis at Stanford are actually successfully getting funding from the NIH, and from their Open Medical Foundation is another big funder of their research. And they're opening up these collaborative research centres, not just at Stanford, but also at Harvard. And a new centre has just opened in Uppsala in Sweden, and they're all investigating different aspects of ME/CFS. So Stanford is focusing on the molecular basis of the disease and of diagnostics and drug screening. Harvard is looking at the central feature of ME/CFS, post-exertional malaise, and taking muscle biopsies of their patients. And Uppsala is looking for small markers, and particularly neuroinflammatory markers in the cerebral spinal fluid. So that is great. Another exciting thing is at Stanford now, but coming to Harvard soon, and hopefully to all us other researchers worldwide, is this open data center. So pre-publication data is going to be uploaded to this data center, and it will be able to be accessed by everyone in the collaborative effort. So that is great to hear. And that is the conclusion of my talk. If anyone has any questions about it, feel free to ask. I rushed through.
Where do you get the money from for your research?
We have had a lot of donations from ANZMEs to fund us in recent years. We've had some success with the Lottery's health grant, but unfortunately for ME/CFS research, the really big 3-year funding grants, they're not interested in something like ME/CFS that goes to cancer and diabetes and the big profile illnesses. So we do rely a lot on just small year-by-year funding groups.
All of it. To operate for a year or 3 years.
I could talk to you afterwards. We're currently looking for funding for next year. So I think we've managed to gather about 30 or 40,000, but we need sort of upwards of 100,000 to really keep the team together and to keep going. But yeah, that's the ongoing struggle, but there's been a lot of generous people and groups that have helped us get this far, so yeah.
Maybe you can tell us about your team there.
Yeah, so there's Professor Warren Tate down at the University of Otago is the leader of our team. His daughter has ME/CFS. She developed ME/CFS in her teenage years and she still has it now in her 40s. So he has always been very interested in the illness and started the research when I joined his team in 2011. We've got myself as a postdoctoral fellow at the moment. Now I've finished my PhD. We have had someone here, Alex Noble, come to hear the talk. She was a master's student in our team. She's now here doing a PhD. We have another master's student just finishing up, Amber. She worked on the epigenetics studies and she's hoping to stay and continue to research epigenetics in another patient group we have, which is a collaboration with Dr Lynette Hodges. So she does exercise study on ME/CFS patients. She gets them to ride an exercise bike and she takes samples before and after this very intense exercise, and then 24 hours after that, and sometimes 48 hours after that, to look at this post-exertional illness. Lynette Hodges, yeah. And we've got actually a medical student with us this year. So she's an honours student. She's completing her honours research in our lab. She's been doing the mitochondrial seahorse study that I showed you a little bit of. So she unfortunately will be leaving us to go back to her medical studies early next year, but at least she'll be going there with a little bit more knowledge and appreciation of ME/CFS. So that's always a good thing.
How do you get people to participate in your studies?
We've been mainly recruiting from Dunedin because we've been usually working with fresh samples which need to be sort of isolated and processed and then used in an experiment very rapidly, but we've been exploring using frozen samples so that we can recruit people from other centres. So yeah. Great.
Do you have anything in your research at the moment that might lead you to trying some type of supplement or something that might be able to give some improvement to some of your
we're looking, as I said, we're looking at MitoQ at the moment. Results still to come. It is a supplement. It does affect the mitochondria. Supposedly it's part of the electron transport chain, and it has been shown in other studies to give really positive effects. The thing we're not sure of yet is what dose might be an effective dose, and it might come down to individuals as well. The dose that they recommend is about 2 tablets a day, about 10 milligrams, and we have had one story of one person with ME/CFS who was taking 6 tablets a day and was having some amelioration of some improvement in her energy levels and things. At the moment there's nothing that we can really recommend for everyone.
Does MitoQ help create ATP or what is it?
It has reactive oxygen species regulatory effects, so it can help dampen down any high levels of reactive oxygen species. And there was the thought that it might, if there was a deficiency in CoQ10 in that electron transport chain, that it might slot in and help sort of replace that. But we're not sure if that's exactly how it works. We can't sort of look at the mitochondria and see where it goes specifically. So at the very least, it has that effect, that positive effect on reactive oxygen species and that oxidative stress.
So did you find that the body was creating good amounts of ATP, and that it was just blocked, or not?
We found some differences. Again, our sample group is small, so we can't say they're significant yet, but we're wanting to recruit more people and to continue. Yeah.
So was the body able to produce ATP?
It can produce ATP, yes, but not significantly, but lower levels than that one patient I showed you, a severely ill patient, was producing much less ATP than our controls.
The Stanford study,
yeah,
where he was looking particularly at complex 5, found that there was a significant reduction in ATP production.
yes.
Effectively, we're running on on one cylinder instead of 4. So
yeah.
Everything else is ramping up, because if we need 3 ATP molecules, we're only getting 1 out. You've got to produce 3 times as much to get in.
Yeah, yeah. Yeah, that was Dr. Paul Fisher. Yeah.
We're running on the spot.
Would it even help to supplement that through particular things to help the body produce it, if it's still being blocked? Like, do you think there could be something in helping with supplements in terms of creating ATP?
I think it can't hurt to try those things. I think ME/CFS is a very complex illness, so just targeting using something, just one aspect of it, will probably help you, but it's not going to completely cure you, and you might get some benefits, and then the effect might fade, or But it's always worth keeping an eye out on the what's available.
Not a cure. It wouldn't be harmful to take it as a supplement.
No, it's not harmful. It is available at pharmacies, so it's there. Yes, 2 people. Would you like to ask first?
I'd like to express gratitude just for all that you're doing, because nothing else, in the meantime, it's providing so much hope. So my question here is, so that's hope from the outside. What can the ME/CFS sufferer themselves do or not do or create to have that hope, to be part of the process of getting better?
That's a good question. I think it's great to be able to join support groups and things that are available, like the group who helped organise this event, because they're great. It really helps to be able to share your stories and to talk about these results and to share the new theories that are coming out. There's some really great forums and things that also keep you updated on a lot of the really exciting research. Health Rising is a really good one, and also the support groups as well. I know you guys have your own website too that will keep people updated. There's ANSMEs, that's the sort of other major national body, ME/CFS support group body. Keep your ear out, because there is a really big push at the moment. I was talking about Stanford and Harvard and Uppsala as these collaborative centres. They're really motivated, they're really sort of pushing. So Ron Davis is the leader at Stanford University, and his son is ill, he's very severely ill, and he wants to help him now, so that's why they're kind of trying to drive this forward, and he's applying for all these big grants, and he's finally getting some success, which is great at getting the funding that's sort of really needed to keep moving research forward. So keep hopeful, because you should be.
Business card on you,
I should have. But I can give you my email address or my supervisors afterwards. Sure.
The exercise study that you mentioned, what was she doing with that? Sorry, do you know a bit more?
She's she's looking at, she's taking blood samples, she's also measuring like heart rate, blood pressure, all those sort of physiological effects. So she's, and she's been sending the blood samples down to us to do the sort of biochemical measurements, molecular measurements.
Is she looking more at kind of the diagnostic side, using it as a diagnosis, or is it?
She's looking at post-exertional malaise as the key thing she's trying to elicit, and then tease out whether it's something to do with the heart rate, the blood pressure, and then also collaborating with us to see whether there's something deeper than that causing those effects.
Where is she based?
She is in Palmerston North, I think. I always get New Plymouth and Palmerston North confused, but I think it's Palmerston. Yeah, yeah. But no, she's also very committed, I think, to the research. She's been going for a while, and we've collaborated for a while.
A few years ago, but
yeah.
How fresh do the samples need to be? I'm thinking, would it be worth it for the Canterbury group to organise sort of kind of a blood drive? Like if you guys set someone up for a day and
yeah, well,
everyone who could was there, and you took our blood and took it home in the same day.
Yeah.
Fly up in the morning, fly back in the afternoon. Would that be fresh?
It's something we could explore. So the seahorse machine that we use down in Dunedin, I believe there's one up in Christchurch as well. So if we could organise it, we could even potentially get someone to come up and do the research here for a time. If we can manage that, that would be great. Otherwise, we are looking at using frozen samples as well. So even if we got samples, froze them immediately, or had someone up here to process them and freeze the cells that we needed immediately, and then could take them down, maybe a few visits up might be needed, then that's possible.
If 6 hours old or 8 hours old it is.
4 hours is kind of what we're working with. So unfortunately the drive.
Flying, but it's
yeah. Yeah. The other thing is it takes about 3 hours to process a single blood sample, so we're limited by our lab technician. Tina is also a very important part of our team, who I should have mentioned earlier. She's the one who does all the blood processing, so she's just one person. So at the moment we think the most we've done at once is 4 people, but she is a wizard. Yeah, but no, it's something we will try and do for sure.
There's quite a few of us up here. Put my hands on probably 4 or 5 severely ill people just on Facebook message. So.
Yeah, well, that's something I can take back to Warren, and we can see what we can do. Yeah.
Do you have an educated guess as to the Stanford study, why there was an electrical impedance in the cells where it was
So one of the theories was looking at I think it was the sodium potassium ATPase and just an imbalance in molecules across the cell, but there's a lot of things I think it could explain it, and they don't know exactly what it is. It's the membrane of the cell, the energy potential. It's just a little blip that they've measured, and yeah, a number of things could cause it. So yeah.
You also, in one of the first slides, was calcium. And
yes.
Yeah, could you just explain briefly what that part, the calcium part, in your role is?
Calcium is an essential for most cellular functions. So, but it needs to be, all calcium magnesium levels are very sort of regulated. So there's lots of membrane transporters that make sure the right sort of concentrations are inside the cell and outside for it to perform its function. So calcium is just one of those very key elements.
Yeah. Is it key for ongoing to get better from it, or is it key just in the just basic?
Basic function, yeah. Yeah. Yes.
The study's looking at
serotonin, that looks like an upregulated serotonin. So were they able to measure serotonin levels in any way to show that that was actually happening? Or
That's what they haven't really done yet. So they've got the Cortene study, is that what you were thinking? Yeah, so the going straight to the drug stage. Yeah. So they're developing this drug already, and they were looking for what it could be useful for. They knew what it did, and they were looking for what it could be useful for, and they looked at ME/CFS, and they have developed this hypothesis, which they've taken to different groups, to different researchers, and sort of presented this hypothesis and said, do you think this seems like a good explanation for the illness? And it is something that's coming up with the Eiren metabolic trap and things, and a neuroinflammation stress response. It is a good theory. Whether it explains everything in ME/CFS, because it is a complex illness and it does have this wide range of symptoms and severity of symptoms, we're unsure yet. So I don't know if it's going to be, the miracle drug that, or even if it's going to be really effective when they actually get to these phase 1 and phase 2 trials.
I'm not really sure if ME/CFS patients have that actual that serotonin at a high level in their...
There's been some studies a while ago that were done that showed lower levels of cortisol in ME/CFS people compared to controls, and some that showed slight increases in serotonin and things, but there's not been that really extensive research that normally comes before you get to the drug stage. So we'll see. The research will be surely, I'm sure people will be doing that now to explore it.
Sorry, one of your slides mentioned in the standard study that they've got a drug that are screening. Is that correct?
Yes. Was it this one? There's 2 drugs.
One is it? Neuroinflammation drug?
Yes, we don't know what it is. They keep it secret. It's got this code name that we don't.
I was wondering if it was going to be a drug for another illness, like a cancer drug. You know what I mean?
Yeah.
Is that possibly transitional?
Well, there was a group that did look at a cancer drug, rituximab, a Norwegian group that sort of was treating a patient with cancer who also had ME/CFS with rituximab, and found that the ME/CFS symptoms were ameliorated in this person. So they had a huge improvement in their symptoms, and they kind of switched from their cancer research focus to looking at ME/CFS, and would this drug rituximab be useful for ME/CFS? And initially, all the phase 1 and phase 2 studies were looking really promising, that it did seem to really help a large group of patients. Not all of their patients, but I think it was about 2/3 of their patients found a relief of their symptoms. And it wasn't until they got to phase 3, because drugs will go through a number of trials at different levels to make sure they're safe and going to be effective. And it wasn't until the phase 3 trials that they found that it wasn't working. Yeah, which was a shame. It was a real shame, but they are still researching ME/CFS, and they've now moved on to looking at metabolism, the metabolic pathways that I was talking about, the citric acid cycle, and the electron transport chain, and mitochondria as a sort of consequence of that, or an endpoint.
There's another one called Epigen that was giving a glimmer of hope somewhere, but I don't know.
Yeah, I have come across Epigen in my research. It is another one that sort of looked it might help some people, but it's not everyone again. And it's
a specific.
Yeah, I don't know if it ever went to like specific phase 1, phase 2, etc. trials for ME/CFS. Yeah. All right. Is there any more questions?
A million.
You can ask me after.
Cool.
All right, thank you so much.
Yeah, thank you. Cool. So I'd like to thank Eiren for giving such an informative talk. Research such as his firmly establishes that there is a biological reason for this illness, I'm sure we've all learnt something this evening, if only that it is a complex illness that is not fully understood. Thank you for highlighting aspects that have recently been explored. So while there is no biomarker for ME/CFS, correct diagnosis and treatment relies on the understanding of the illness by the practitioner. So to provide more comprehensive information, our group, the ME/CFS Group Canterbury, has been working with the CDHB over the past year to update the Health Pathways page on ME/CFS, so that should be available in the next couple of weeks or so. We have seen the final draft and I'm pretty happy with it. It's just they won't give us an exact date. So we are also working with the CDHB on developing some online continuing medical education for GPs, and we're hoping that that'll be available sometime next year. So for those of you with patients with ME/CFS, we are developing a referral process for our services, but it's not up and running yet. We understand that treating people with ME/CFS in the current primary healthcare environment is really difficult. You only have 15 minutes per appointment. So as the community facilitator, I provide individual support to people with ME/CFS. So usually that involves visiting people in their homes and discussing with them what issues they're having and how they might manage that. The main management strategy for ME/CFS is avoiding triggering post-exertional malaise. So a lot of the work that I do with people is around activity management and how to avoid triggering post-exertional malaise. So the ME/CFS Group Canterbury does have a strong focus on peer support, and we run monthly group meetings so that people with ME can get together and share their experiences and learn from each other. So lastly, I'd like to thank you all for being here. Knowing that there are health professionals who are interested in learning more about ME/CFS, provides hope for sufferers that there can be some improvement in the treatment of ME/CFS in the near future, and we hope to run similar events in the future so that people can continue learning. If you have any questions about ME/CFS in general or what services we provide, then please get in contact with us. Thank you. So thank you all for being here, and that's about it, I think.